Alright students, in this video I'm going to go over the basics of transcription, a review of transcription, and an overview of translation. So what does this have to do with our class? This has to do with what's called the central dogma of life, central dogma of biology, central dogma of biochemistry, which simply it's DNA encodes for proteins. The information of DNA is copied into a molecule of RNA. It's also called messenger RNA. And that messenger RNA is translated into a chain of amino acids which make up proteins. That is what the central dogma is. It's a very important concept in biochemistry, which is required to understand in this class. So I'm going to go over transcription really quick and then translation. So here we have our cell. It's a eukaryotic cell. We have a nucleus. We have the information, the blueprint, the code for proteins in DNA. There is a process called transcription, and that's what the gizmos activity A was. The information is copied into a molecule of RNA, and then the RNA goes outside of the nucleus where it's translated into a chain of amino acids and makes the protein. So that's what we're going to go over today, translation. And there's this process happening right here, and we have very important structures called a ribosome and tRNA, which called transfer RNA. They're the ones who bring the amino acids to the ribosome, and they are paired with the codons, the three bases at a time of the mRNA. So that's what I'm going to go over right now. So right here we have a DNA sequence using the base pair rules. C goes with G, A with U, C with G, and so on. But translation is where now the amino acid, not the amino acid, the mRNA codon, which are three bases, a codon is three bases, that is going to be translated into a protein. And there's the bases are going to be translated into an amino acid, which make up protein. So each of these letters represent an amino acid. So sequence of DNA, sequence of RNA, sequence of amino acids. Here is, I've talked about this before, the goals is to translate an mRNA codon and explain translation. But right here, we have the technology that is used to make the COVID vaccine. mRNA, a synthetic mRNA is used. It's injected into the muscles, and the ribosome, which are responsible for making the chains of amino acids of proteins, make the proteins of coronavirus, and body builds immunity to it. So it's important to know because it's relevant. The COVID vaccine uses mRNA technology. So right here we have the RNA polymerase and made the copy of mRNA, and then the ribosomes are what going to make this whole chain. It's also called the polypeptide, but for our class, just know it's a chain of amino acids, which is a protein. So right here it says translation is the process of translating the sequence of mRNA right here, mRNA, here's a sequence, into uh, a sequence of amino acids during protein synthesis. This genetic code describes the relationship between the sequence of base pairs, the ATCG, in a gene and the corresponding amino acid sequence that it encodes. So basically what that means is your DNA code, which we all have DNA code is what's going to be translated, and the DNA code determines the RNA code, and the RNA code determines the protein sequence. So ultimately, the source of the information is in our DNA. That's what holds the genetic code for proteins. So that's what that means. So right here we have an amino acid sequence, and then here we have the codons. Codons are three bases, three letters, A, U, G. That's going to make a amino acid called MET. These are abbreviated by three letters, but they have full names. Methionine, we have ACG, and that makes I think that's called tyrosine. I'm not completely sure from the top of my mind. Uh, you know what I mean. So we have JAU, and that makes ASP. So do you see the code of mRNA is making a chain of amino acids? We have a codon, AUG. Methionine is the start amino acid. Basically, what that means is the body or ribosome knows, if it was conscious, that that's where the amino acid sequence is going to begin. And then we have a stop codon, and that stop codon is what stops the amino acid from growing, and then we make our protein. So start and stop. Your body is like a little factory. Some switch. The switch for on is AUG, and the switch for off is UAA, and then we have our entire chain of amino acids to make a protein. Each group of 3 bases is mRNA is called a codon. Each codon corresponds to an amino acid. Proteins are chains of amino acids. So let's look at an amino acid sequence. So right here we have an amino acid sequence of insulin. Insulin is talked about for diabetics. Diabetics don't produce enough insulin, or they don't respond to it. But look at all of these amino acids bonded together, and then we have other types of bonds right here, are what make up the protein. So here, another example: DNA, RNA, RNA. Um, RNA determines the amino acid. Then we have these structures called tRNA. tRNA carry the amino acid to the ribosome. So basically, what happens is the mRNA is read by the ribosomes 3 letters at a time, and then the transfer RNA goes to each codon, and there's an anticodon, and that's what follows a specific pattern: A with U, A with U, A with U, A with U, G with C, C with G. But for our class, you need to understand that the transfer RNA is what brings the amino acids, and it's paired with the codons on mRNA with an anticodon, and it's done 3 bases at a time. So it's kind of like a little machine, and I'm going to show you a video during class so you can visualize what's happening. When it comes to the main functions of ribosomes, they assume the role of bringing together amino acids to form particular proteins, which are important for completing the cell's activities. So obviously, I didn't write that, hint to what I talked about in class, but that's the textbook definition. And basically, what it means is these ribosomes are what build the protein. You have to be, I never liked this part of the lesson, but you have to be able to translate amino acids, and you have to use this really strange table, which we're going to practice together, how to decode or translate the mRNA codons. So basically, for example, you have these on the gizmos, part B. What amino acids do the following codons code for? So how it works is, see, I don't like doing this on the computer. You have to do it on paper, but you have to go 1 letter at a time. So AUG. All right, so let me see if I can do this. AUG, that means that you're going to start, this is the first letter right here. You're going to start right here. So I'm holding my cursor over here. And then I'm going to go to the second letter. The second letter is U. So it's A, and I'm going to move my cursor until I get to the U column. Oh, I'm already there. A, U. First letter is A. So, okay, percent, I don't even know where to begin. All right, A is the first letter. Okay, so I go here. And then U is the second letter. So I go here. And then you have to go to the third letter, which in this case, it's G. So I'm right here in the beginning. A, U. Let's go down to G. How did I get there? Oh, look across, third letter, AUG. AUG stands for MET. MET is methionine, and that's the amino acid that's referred to as start. So AUG is MET. Sometimes they write it as met, but let's stick with met. Met is the first amino acid. CUG. How do I do that? So, okay, I'm starting right here. C is the first letter. Then I'm going to move my cursor or my finger uh, to the second letter, which is U. C, U. I'm already here. And then CUG. So CUG, I move it right here. CUG. How did I get there? CUG. CUG is right here. CUG, and it's right there. LEU. LEU. That's leucine. So what the body is doing is the ribosomes are reading the codon one codon at a time, which is 3 bases, and then the amino acids are brought by the tRNA and they're matched up, and they connect together and they form a giant protein. Some proteins vary in size. Some proteins are small, some are big. It all depends on the sequence of DNA. So we're going to practice this in class, but I wanted to show you how I got it. If you want to pause the video, you can get a scratch paper and try and see if you can do this one by yourself. So ACC. So pause the video right now and do ACC and then see if you got it right. So, all right. So you pause the video. ACC, A right here. ACC is TRH. All right. Hopefully you got that right. This is just another example of a diagram of the tRNA. tRNA bring the amino acids to the site of the ribosome, and anticodon matches with the codon, and then these amino acids are bonded together. I think once we see a video and of actually happening and this video too, it's going to be easier to understand. All right, that was too long.